which is, as you know, that DNA markers are very much not affected by the environment. Then here I have to go only the study of some of the things like marker-studded backcross breeding, which is very much important in vegetable as well, particularly for developing the different resistance line with respect to in the crops like tomato, the cucumber. Many people have worked in the worldwide, but in India there is very, very less work has been done. So that already you have know about this marker-studded backcross breeding. I will not go and touch upon that. Gene pyramiding is also very important in vegetable, as you mentioned about this success story in case of tomato. If you know the tomato, earlier we don't have a varieties which is resistant to tomato leaf curl viruses. But now we have some of the varieties have evolved in 2017 onwards. Some of the varieties are really good, particularly in case of Arka, now the multiple resistance variety. In case of your cucumber also, some of the varieties which is resistant to powdery mildew also developed for this marker-studded breeding. Gene pyramiding, as I mentioned, that is done in case of some of the crops, mostly in case of tomato and cucumber. But in India, it's very concerned. The marker-studded breeding is very infant stage. Nobody has worked on this particular crop, but it can also work for this early generation marker-studded breeding, by which is very important for us to develop the new lines of the new varieties with the different type of quality attributes. Now, the marker-studded breeding cannot be done alone, because if you go for only one time of genotyping of the plants in the F3 or F5 or maybe in the backcross populations or BC2 or BC3 populations, you cannot get some type of successful in different traits. So you have to combine both the markers as well as the phenotypic selection. So sometimes both the things are to marker-studded selection as well as the phenotypic selections we have to go for simultaneously, then only you can get the improvement of some of the vegetables. So that you have to get maximize in the genetic gain, and level of recombinations between the QTLs and the markers will be more, and reduce the population size if you go for marker-studded selection. So these are some of the benefits you already know. Now we come to the example where we have the dent on das, particularly for this marker-studded breeding in tomato, which is, you know, that most of the cases is started in Aviar DC, that is in Taiwan. They work on the different species of marker-studded breeding in tomato, and they use different species like Solanum herbaceatus, Chileans, then Solanum pimpinellifolium is very important for us, which is actually important for vitamin C content, as well as there are resistance to the tomato leaf curl virus. Similarly, in case of lateral blight resistance, in case of Solanum, that is in Solanum pimpinellifolium is very important. So they have their genes already there in the wild species, though they introgressed in the different lines of Solanum lycopersicum. So that is there as Clinton. Now, in this case, here is some of the study, I already mentioned that some of the studies work in IHR, some of the study work in IAVR, and some of the work is also studied in IARI. So that only I will give the example. Other things you can read it from books which is available to you only. Now this is the methods we go for screening. So screening is very important for any type of disease infestations. If you go for phenotyping is very important. No, if you are not going for phenotyping properly, I don't think you can have a better understanding of the genes or better understanding of your marker-studded selection, which is very important. So here we people have started working on this tomato leaf curl viruses, that you know that 3 types of viruses already exist, more than 3 types. There will be 9 or 10 types of viruses are there in different strains like Bangalore virus, New Delhi virus, Palampur virus, now Raipur virus, Jaipur virus. So there are a lot of viruses are there in case of strains in case of your tomato, which is very important or challenging for us to develop the new lines. Okay, so these are the methods of inoculum by which we can go for this phenotyping of this, particularly for the tomato leaf curl New Delhi virus, which is mostly, as you know, that we have 2 types of viruses, the strain type. One is monopartite and another is bipartite. That is already, I think you know, you might be knowing, those are working. So monopartite, it is generally seen in case of South Indian condition, that is in Bangalore, you have monopartite virus, here is the Bangalore virus. But in case of New Delhi virus, it is a bipartite type of virus. Okay, so that depends on then the genes we have to isolate it for the different species, also totally different. So that is, as you know, that there are 6 genes are now already there in tomato with respect to different species concerned, TY1 to TY6 genes. And TY1 and TY3, they are mostly similar, but in case of TY2 and TY3, which is very, very important for us to develop against the tomato leaf curl nudilly virus. So it is very important for us. So you should know on that, particularly in case of tomato. So there's you have to assess the diversity based on or the severity of the different species or your lines. So you have to screen properly. Their symptoms also you should know. What type of symptoms will be there in case of viruses, and based on that you have to identify. Also you have to score them, which is very important. Now the strategies are totally different. So two strategy, we can go for backcross scheme or we can go for cross scheme, as you mentioned. I already taught you by different crops also. Here also the same thing also you can apply, particularly for the backcross scheme. We have to develop the introgress lines through this particular or gene pyramiding with respect to the backcross breeding in case of tomato. Now if you see the screening methods, you have a variety like Panjachuara, which is highly susceptible. So that you can see here, and you can have a different strains of viruses like tomato leaf curl Bangalore viruses. Here we have TY1. We have when you incorporate the introgress that particular genes, we have some resistance lines you can develop. But we can have this TY2 or TY3, then you have a combined effect of your both monopartite and biotic virus. So that is very important for us. So they have screened many of the lines and also introgress the lines with the simple way of marker-casted breeding. And already there are a lot of markers already developed in this particular aspect of TY. All the genes is concerned from T1 to T6. Already the genes or the gene-based markers have already been developed in respect of this particular tomato leaf curl nudilly viruses. So that already I mentioned that screening procedure, and then the next one is that they make it some hybrids by which suppose you have some inbreds. So you can also develop like Orca Aved, if you might have seen that the background is totally different, the lines are they will totally inbreds they develop is totally different. Then cross it, and both are showing resistance to different diseases. So the simple way of process of by making a hybrids also with respect to one particular disease resistance, another resistance, and you can make a crossing, you can develop the multiple resistance in the hybrids. So this is the very important which is done in case of Orca Aved. Then many of the cases also people tried in IAV here, like Kasi Aman they developed, which is also resistant to tomato leaf curl nudilly viruses. So Kasi Aman was a very good varieties in particularly in the northern belt. So they used to have that's materials or the source material they got it from the Aviar DC also, and they tried their own variety like Kasi Aman to develop these resistant varieties in this respect. And also they also incorporate or introgressed the lines of TY5 genes, so and then combined the totally resistant type of lines they developed in this particular tomato crop. Now in IARI also we have used different donors with respect to the genes related to bacterial resistance, with respect to lead blight, with respect to the tomato leaf curl nudilly viruses, CMV, so cucumber mosaic viruses. So that grey leaf spot, heat tolerance and all things are there in case of ABR-DC lines. And many of the people are working for introgressing our own variety. In case of IARI, we have a lot of different varieties we developed, and now we have started for introgressing these lines with different resistance to different biotic and abiotic stresses. So that also did the genotyping in the population, and we have also developed some of the F6 advanced generation, some of the lines which is really performing well with respect to the tomato leaf curl nudilly viruses. So that is there. Already some of the lines are in the trial. Now coming to the another crop is a cucumber, which is a very important crop for me because I started here in cucumber as also in bitter gourd. Most of the works also I worked in the University of Wisconsin, that is in USA, and some of the works also we started here in working here. Now gynosis line is very important. Gynocism is important in case of vegetables, because if you have gynocism, definitely you don't have to go for that hand pollination for making the crosses. Okay. That is like your male sterility. Today you are talking about male sterility. Similarly, the gynocism there in case of cucumber, as well as the first time we developed this gynosis line in bitter gourd. And here there is no need for any pollination. You have to, because many of the people or the farmers or this private company, they have to go for the pollination by hand, which is really very cumbersome as well as very labor intensive. That's why if you have a gynosis line in one female parent and male parent to grow simultaneously, there will be a crossing by the insect and you will get the hybrids very easily. So you can reduce the cost of productions of the hybrids. So it is very important in case of both cucumber and as well as in case of your bitter gourd. Any cucurbits is very important for that. So in that case, in generally in case of lines what you have, the gynosus line is developed in the western country, particularly G421 and GY14, which is in cucumber, which is having the gynosus line, but there are only drawback is that in case of Indian lines is totally different from the your lines of the western countries, because they have used for the pickle purpose, and they have to go for only single harvesting. But here we have to pluck the fruits, then new fruits are coming, then you can also have more number of branches, and all things will be there. And another important thing is they have a dark green color of cucumber, but we prefer to have a light green cucumber varieties. So lateral branches will be more in our case, but lateral branches will be very less, at least 1 or 2 may be there, or there will not be any lateral branches, because they have to go for mechanical harvesting. But we are doing for the plucking or for the slicing cucumber. And we have a slicing cucumber and or salad purpose, and they have a pickling cucumber for pickle purpose. So these lines I got it from US, which is GY14 and GY421, which is having these characters, which is having only single or only one, there are 1 or 2 branches, not more than that. But we have a branches about 4 to 5, 6 branches are there in our own Pusa Nabeen, Punjab Nabeen, and Pusa Barkha, and some of the varieties of Pusa Uday. So these we have to integrate, and that schematic presentations I made it here, that you have to go for in BCU and backcross population, and then develop this Markasted selection, by which you have to go for foreground selection. Then by the foreground selection, we can develop the lines or the materials, and then you go for background selections with the different markers. And we have taken the lot of markers, particularly for background selections, and these markers, those who are used particularly for the, as it is cucumber genome sequence is available. So we use the markers which is known markers, which is means linked to the target traits, particular lateral branches. It is already there. There are 4 QTLs are there, which is already in the public domain, which is there. We have the growth of the plants. So vegetative growth, maybe the single branch or 2 branch or 3 branch or highly vigorous. So that also markers have been QTLs have been already reported in case of your cucumber, and there is no problem to get this marker. So based on that, you have to select the foreground background selections also. Not only the foreground selection markers are already established, but if you have the markers, particularly for the different traits in the quality traits in case of cucumber, then definitely it is also available. So we have to choose the markers really which have having the link to the trait of interest. Then only your programs will be speedy. Okay, otherwise you have to go for a long time. You have to see that which are the polymorphic markers, then you have to go for a huge number of markers, particularly for the background selections. But here there is no need of having huge number of markers for the background selections. If you know the markers for the target traits, the quality, like fruit length and diameter ratio, LD ratio, that quality, the seed cavity thickness, all the markers are available in case of cucumber. So based on that particular that sequence available, you can also take your own markers, and SSR markers are already available, so that you can utilize for your even for the background selections. So that is important, and we did it the same way, and we also developed the number of inbred lines with respect to this one, and we also have a very good response to that is recurrent genome recovery in respect to the different crops and different varieties, and we can find the improved Pusa Uday, improved Pusa Panthkhira also we developed, which is monoecious. Earlier it was monoecious line. Monoecious means there are a lot of female flowers, male flowers. That is 30:1, 30 male flowers and only 1 female flowers. Now we converted them 100% gynoecious, that is 100% female flower. As I mentioned, this female flowers is more, definitely the productions and productivity will be more. So that way we have definitely the hybrids also, the combinations will be there, and definitely we have predominantly gynoecious hybrids we can develop, which is having more number of female flowers, by which you can have a productivity of the per plant, productivity will be more in case of cucumber. So that is our most important things we got it from this one. So now we converted our own lines to this gynoecious line based on the Markasted selection, and within 3 to 4 years, we developed 4 lines of different background with respect to the different varieties. With respect to the Barkha, which is also resistance to tomato leaf curl viruses and CMV. Now this tomato leaf curl virus is there in every crop, and if you see these vegetables like tomato, if you go, go for all the crops of cucurbits also, now also rampant with this tomato leaf curl viruses. So some of the lines also we developed with respect to this one. Another important is target leaf spot resistance in case of your, this is in also cucumber, also you work in this one. So the important is that you have to go for standardizations of protocol, particularly for the pathology point of view. But I am not a pathologist, but we have developed some of the techniques we have through which you can go for rapid screening of the methods of for this target leaf spot in case of your cucumber. So that also we developed. Then we have seen that some of the lines we use, that is Wisconsin lines, there are US lines are there, and we also use the growth chambers. If you have growth chambers, then it is very easy, or the simple technique by which, because they need a particular specific temperature, humidity, particularly for the growth of the different fungus and bacteria also, that you know. So if you have that particular standardizations of the techniques, it is easy for screening. And if you go for field screening, that you know that you have to have all sort of things and a lot of space, a lot of materials you have to develop. But here what happened, you have to go for only in case of your small chamber, that you can go for 100 plants of screening. You can do it easily with respect to fungus or bacteria if you are concerned. So that also we did it. Then we also perform the inoculations methods when you have to go for inoculate them, either in the cotyledon stage or in the broadleaf stage. So based on that, we also inoculated them, and we can also screen them, and we found many of these elite lines which is also resistance to this in case of your target leaf spot. So then you classify them. Based on that, you have to score them. Based on the symptoms occur, so we have to score them, and we can also develop some of the lines with respect to the resistance to this target leaf spot. And then we score with this F2 derived F3 populations. We have to go for phenotyping, and also we can do it in the chamber. If you have a chamber about 400 to 500 plants, you can screen very easily. Okay, so you have to have one tray, and that tray more than 50 plants you can grow them. Then you can maintain the humidity by putting some of the plastics cover, and you can also maintain the humidity inside and the temperature inside in a growth chamber. So 300, 400 plants very easily within 1 month you can screen 300, 400 plants in this particular chamber. So that is based on that you can also see that. But another important thing is that the genes, to get it, the source is very important. And here we got it different things. Here we got the source. Some of the sources, when the background changes, you have the genetics of this inheritance also changes. Some are controlled by the single gene. Okay, if we change the background, and that is there, and some are having dominant genes, and some are also recessive genes. That depends on the sources from where you got it. So based on that also, you can have multiple sources of resistance with respect to the particular diseases. As it is in case of a target leaf spot, also you can get it in different crops in that particular way. So we did it that one, and then we go for QTL mapping, and we can find that in case of chromosome 6, some markers which is directly or flanked to this target leaf spot disease. Then in other populations, I mentioned about this 9930 and 7012. These are the different backgrounds from which we converted our lines to the target leaf spot resistance, and we are also getting the dominant traits in this respect. So I will not cover this one, but the identification of markers is really very important, particularly for the different genes, particularly for the recessive genes. If it is controlled by that one, is very important. If a dominant, it is not so much of important. But for background informations, if you go for background selection, if you have dominant traits, it is also very easy to get, particularly in case of your gynosis is concerned in case of cucumber. Now we also did the association mapping with respect to the powdery mildew resistance. As you mentioned that there are a lot of markers has been generated or mapped, particularly for these particular traits of powdery mildew, but still today they are not stable so much because the resolutions is not so enough, particularly for if you go for biparental mapping system or through the markers restrictions is not so easy. And also you have to go for QTL identification, it's also totally different. In some backgrounds it is giving different story, and some other backgrounds it is giving different story. So it is very, very difficult to have actually the QTLs which really to identify them through this marker. So now we have to develop their own techniques, or people also tried with some of these, particularly GWAS is very important nowadays, and people want to go for this type of experiment with the GWAS experiment, and they got some of the alleles and identified different genes with respect to, in case of your, and there you can have a true pictures about this background of that particular lines, okay. So that I think you know about this GWAS, it is already been taught, no, GWAS. So I don't think it will be important for me also. Here also I already mentioned about this scoring for phenotyping is very important. If you are not scoring properly, as I mentioned, that is totally misnomers. You will not get a true picture or true QTL identified with respect to the marker selection. So it is very important for us. But in India, you have very much of, not so much of inputs are provided to the universities as well as the different institutions. It's very, very difficult for a particular screening of the material. We have about cucumber, about, as I mentioned that cucumber is originated in India. So we have about 400, 500 accessions are there in our gene bank. Those are not repetitive, but still we are not screened about 100, those are not repetitive. It's very, very less amount of materials or genotypes we screen. Similarly, in case of bitter gourd, in case of any other crops if you are concerned, many people from here, when we have the cucumber, in 1994, the people from US they came here and they took cucumber germplasm, about 600 germplasm from India, and we lost all the germplasm, but they have there in the introductions, plant genome introductions, they have already put it there about 600 accessions, and most of the genes they work from these Indian genotypes. If you say I work there in US. And when I came here and I searched for this introduction, I have to get it from US. So this is the very peculiar thing in India. We lost most of our germplasm. If your germplasms are not there, what to do with this resistance breeding? What to do with the improvement program? So many of the things we have to conserve ourselves, which is very important, that you are not doing. So in case of melons, if you have only one introductions, they have a fusariumable resistance, they have virus resistance, they got it in US. So now we are procuring that materials for us for the improvement of these melons. So that is another painful thing for us. But anyway, so these are the materials you have to screen, and that way you can also score them, which is very important, and scale them. Then the genes also have a different things in the cotyledon stage. The genes are different. In case of your full true leaf stage, the genes are also different. Even for the hypocotyl also, powdery resistance genes are also different. So in that case, you have to screen properly, which I mentioned is very important, both in cotyledon stage, in hypocotyl stage, also in the true leaf stage. That also we screen it properly, and then we go for Snp genotype. So if you can see that particular locations, you can find that in the same tray or the different tray. If you put it in the one chamber, you can find the variations in the level of infestations of this powdery mildew in case of cucumber. Now these lines also we are utilizing, particularly for resistance to this powdery mildew resistance in case of cucumber. And then another important thing is that Upal, you have time, no? No problem. It's okay. So now this marker backward selections also we did it in case of cucumber to develop the new lines with respect to some of the lines of China. That is cucumis hystrix, which is very important lines, which is resistance to powdery mildew. Also a lot of multiple branches, also sequential fruiting. So that also is used here. Then we have scoring them based on the vigorous plant growth. We have already known markers, particularly for the growth of the plants, and the QTLs known for this lateral leaf branches, which are used to develop the new lines having the sequential fruiting habit, also resistance to the different nematode resistance, the target leaf spot resistance, and also powdery mildew resistance. So another important crop is in cauliflower. So that also we did it in here in IARI, that is particularly for OR gene. You might be knowing about the OR gene. As is golden rice, we have that golden rice is there. Similarly in case of cauliflower is very less amount of the beta carotene, about 0.5 microgram per gram. But if you have increased from that particular cauliflower lines, which is taken it from California Davis, they have a natural mutant lines from where we got this beta-carotene Gourd gene, and we also incorporated through this Marker-assisted breeding. Now we have about the lines which is called as Kusa Kasari Beta 1, which is a variety of, in case of your cauliflower, which is having 10 microgram of your, this one, that beta-carotene per gram of your edible person. We also work in capsicum, that is the most important disease in the leaf called viruses. So that is also important. So we have some resistant lines, and we map them. Also, we develop some of the, we have also the information about the SNPs, and then with that SNPs we are going for developing the PCR-based markers. I am quick because I have to, we have some programs, so sorry for that. So it is in the last, but it should be given a 1 hour, then only you can have a lot of things are there. Another important crop is a bitter gourd, which is close to my heart. I worked for 22 years in this bitter gourd, and that is very important for us. Then we have our own lines in case of bitter gourd. Only 5 minutes I'll take, okay? So that lines already we have given to the private seed companies based on this commercialization, that is Ankur seeds and the Namdhari seeds, and they develop, they take it, that one, only to minimize the cost of production, particularly for this one hybrid seed productions. Okay, so that they took it from us, and you can see this bitter gourd. If you one side you can see that how many male flowers are there, but another side you can see that only the female lines will be there. So it is totally different, and with that lines we developed. And as I mentioned, that why I am telling these lines. This is the lines which developed by one of my student. He is also working as a scientist with me, Dr. Dey. He was working with me, and that time, first time we are just in the field, and we got that line. So as a student, very important to go for phenotyping. So that is, if you are not doing in the properly in the plant itself, you cannot get it. So that plants, now we have some modification. Then that was a small type of variety. So we make it by the repeated backcrossing. We make it a medium varieties, and that we are utilizing for developing the new hybrids. And then we also did it the first time the QTL mapping with respect to different traits in bitter gourd. So the one of my students also work on this QTL mapping in this bitter gourd, and we have already published in the Gourd journal. And this is the information or identifications of the QTLs with respect to different things. And gynesis trait also I already mentioned, which is the flank to the with the gene of interest is a very tightly linked. And then now we are starting for the mapping of QTLs, particularly QTL sec methods for the trait like fruit length, tubercles, and other traits in case of bitter gourd. Because bitter gourd is now very important for us for the medicinal point of view, and we now we're working for these different QTL sec methods, QTL secs for different traits in the mapping of these traits and go for marker-assisted selection. And also we started also for SSR markers. We did it with the tubercles, and we have gene of interest, but we are going for the fine mapping, particularly for developing or the link-based markers with respect to the this one. So we will have a lot of populations with respect to different bitter gourd. About 117 core collections I have throughout the world, and that lines are totally different. And if you have the Indian lines, it's different from the Chinese, different from Taiwan, different from US lines. So the totally based on the morphological characters of the fruit shape and size and color and everything will be different. In India, you might be knowing that in case of if you go to south in Kerala, they need to have a white type. So we have that white type, pure white. There is a pearl white type. So that lines is very important for us. So that we are also going for improving the different traits in respect to this one. And you can astonish. I have screened about 117 lines of bitter gourd. The amount of anti-diabetic properties like charantin, saponin, your momodicins are totally different from one lines to another lines. And we have a very small fruited lines, which is very much of 70 microgram of your charantin, and compared to the other long fruited lines, about hardly 5 to 6 microgram of gram weight for fruit types. So that type of things, contrasting characters, we can have also used for our material to develop that lines having high content of these anti-diabetic properties. So that also we are utilizing. Similarly, in case of your bitter gourd, as you know that you use only in the green stage, but if you ripe stage, nobody using. Okay, so that also very important. But ripe stage, it is, we have seen that the content of the nutrients also different from the fresh stage to the ripe stage. If you see the anti-diabetic properties, it reduces when it goes to ripening. But the antioxidant like carotene, lycopene, they are increased when you go for ripening. So in Africa, nobody eats in the bitter gourd in the green stage. They eat only in the ripening stage. And ripening stage because they are very sweet in taste. But if you bitter stage, it will be bitter in taste. So we developed some of the lines also in green stage. They are very much of sweet. Not sweet, you can say less bitter. That is, we have one Pusa Rasdar variety, which is, you can see here the small variety is again here in the left hand side. You can say the Pusa Rasdar, which is grown only in the polyhouse, which is less bitter compared to the bitter gourd what we have in the Indian type. And that is having very good yield if you go for growing in the net house conditions or the polyhouse conditions. So with that also we validate the markers with respect to the tubercles with the different germplasms of the bitter gourd. And we also have these things, but the impact of mass in vegetable crop improvement, they have not started still today. Like that of other crops like rice, wheat and other people, they have worked many years, and many scientists are involved in respect to this particular trait or of in different crops. But in vegetables, vegetables are very high, about 117, 120 vegetables we are growing in India, out of which only 35 are very important. But the working, if you say the work in the improvement program, only 2-3 institutes are working in the vegetable improvement, maybe IHR, maybe IAVR, maybe IARI. But I think the people are doing mostly in the conventional way. But when you make a cross, you have a large, huge amount of population. Even to get these genes stable for this particular trait also, it is very important if you have a huge population. If you go for only marker selections, then only you can have bad or good materials to be selected for that particular purpose. So that resources of the mass is not available in vegetable crop improvement. Then genomic information is also not available. I already mentioned the very few people are working in that. Then inoculation, the QTL mapping is also not done properly with respect to, we don't have a proper networking projects in the case of vegetables, like that of rice, like say wheat or maize. There are a lot of networking projects throughout this country is going on. But if you have a network, actual QTL mapping can be done, and we can definitely have a better marker selections we can do in the vegetable improvement. Then QTL's effect that also depends, I already mentioned about the genetic background. We have a different genetic background, definitely the QTLs will may be different. So that also we have to take care. And lack of markers in polymorphism, because we are not broadening our materials. We are losing our materials, but we are just narrowing down our materials, but we are not broadening our material. So there are a lot of future challenges for us, particularly for you people. If you are, okay, you are now in maybe in some of the crops, but when you go for a scientist, maybe you can be appointed in the vegetable breeding or vegetable crop improvement. So you work on that particular aspect, I think the future will be very good. And your involvement, particularly for the students, is very important for us to have a new lines. And I must thank to my own students. I had